So very honored to introduce Professor Pei-Yong Shi, who's the director of the Institute of Drug Discovery at the University of Texas Medical Branch. And as you all probably are aware of, Professor Shi has a long-standing interest in antiviral research focusing on illuminating mechanism of viral replication and pathogenesis, and how this can be applied in the development of novel diagnostics, vaccines, and therapeutics. So please, Pei-Yong, the floor is yours.
Thank you, Peter. Thanks everybody for taking the time to join this call. And I think Dr. Malevich gave a very good overview of where we stand in terms of understanding the disease and epidemiology, and then also the potential therapeutics. So as she very clearly indicated that after the infection by the biting of the infected mosquitoes, 25% of the individuals will have symptoms. And unfortunately, a small portion of them will develop into severe diseases. And that is mainly dengue hemorrhagic fever and the dengue syndrome. And then some of them will lead to unfortunate death. So the idea really for therapeutics is to prevent the infected individuals from developing into severe disease. So now, if we go back 20 years more, 25 years of the dengue drug discovery, we have the field has been really pursuing three different directions. The first is directly targeting the viral targets, and that is well-proven antiviral approach. And then the challenge of these approach is a narrow window of the time of treatment, as Nalika already clearly mentioned. The duration of the viremia in the infected individuals is pretty short. So the narrow window of the direct antiviral treatment is a challenge. And then also we know any viral treatment will potentially lead to emergence of a resistance. And that is something we also need to be aware of. So that's why I think multiple mode of actions of the inhibitors that will be needed to overcome this limitation. And then the last one for this approach is really, it is not easy to have a compound that can inhibit all the 4 serotypes. And there are a lot of literatures that indicating that some of the compound can inhibit 1 or 2 and not all of 4 serotypes of the dengue. So that is the main challenges if you review the field. The second approach is really targeting the host factors. And we know the virus rely on the host to replicate and to cause disease. And so that is a very good approach. The advantage of that is the field believes that it will reduce the resistance, it will mitigate the resistance issue, as I mentioned before. And then the challenge of this is really the toxicity of targeting those host factors, because sometimes you also trigger or interfere with its physiology function of that host factors. So, and if time permits, I'll also give you an example. Some of the experience I had that some of the project against target, getting to the vivo, it could also be challenging, even though they work very well in vitro. So then the last approach, which is more a holistic approach that will allow us to block the pathological pathway, right? So that will give us increase the treatment window. Time of the treatment will be improved. And then, but the unfortunate, the reality is we still lack of knowledge in terms of the molecular details and how these pathways was triggered and then lead to the severe disease. So that is just pretty much overview of where the field stands and what the effort has been made in the past 25 years. So now, okay, so just the real quick, the dengue, like all the other flaviviruses, it contains a genome of a single-stranded RNA of plus sense polarity. It has a single open reading frame that encodes a very long polyprotein that gets processed into 10 mature viral proteins. At the end terminus, it has 3 structural proteins: C, capsid, membrane, and the envelope. They form the particles of the virion. And then at the C terminus of the polyprotein, it has 7 non-structural proteins indicated by NS1 to NS5. 2 of these NS non-structural proteins has enzymatic activity. NS3, it has the protease, helicase, NTPase, and other activities. And then the NS5 has the methyltransferase activity at the N-terminal domain, and then the 2/3 of the C-terminal domain of NS5 functions as the RNA-dependent RNA polymerase. And most of these protein structures has been solved, and that provides a lot of modern platforms for the modern. So now I'm going to just, if time permits, I'll give you 4 examples of the project I went through, and I think this very representative of different antiviral approaches that have pursued in the field. So first is the nucleoside inhibitor. As we know, nucleoside, nucleotide really form the cornerstone of the current antiviral therapy, right? So when I was at Novartis time, we pursued, looked at all the available, the nucleoside nucleotide inhibitor. We identified this compound, adenosine analog called NITB-008. And now this compound has become really the tool compound for any flavivirus research. So compared with the Y-type adenosine, it has 2 modifications. The nitrogen here is changed to a carbon. And then also the 2' position has a acetylene group here modified. So that's the 2 modifications compared with the Y-type or the natural adenosine. So this compound is not toxic in cells. Whatever cells you use, it's not toxic. It inhibits dengue, West Nile, yellow fever, Powassan, which is a tick-borne, flaviviruses, even hepatitis C, with pretty potent sub-micromolar EC50. And it's very specific to flaviviruses, doesn't inhibit other viral family members like a VSV, E. encephalitis virus, the alpha virus. And then it shows a very good PK as indicated here. In the interest of the time, I'm not going to go into the details of the parameters. It's very, very decent drug-like profile of the PK. And also, because it's a nucleoside, you wanted to convert them to the triphosphate before it can be taken by the viral polymerase to function as, in general, an RNA chain terminator. So you can see if you dose the rats, the rat plasma, you can see if immediately after dosing, you can see the parental compound. And then afterwards, you can see in the red blood cells, it gets converted to the monophosphate, diphosphate, and the triphosphate. That really indicates that it metabolites to its active form, which is triphosphate form, that potentially can be taken by the viral RNA polymerase. Now, in vivo, we use the AG129 mouse model to look at in vivo activity. Let's focus on the middle panel here. First experiment we did is the immediate treatment. So using a dose escalation. So you see 3 mg per kg to 50 mg per kg. You can see a very dose response antiviral viremia activity is model. And if you started to delay the treatment, so the viremia was measured on day 3. If you started to to start the delay, the beginning of the treatment, you can see 12 hours all the way to 48 hours delay. And then, as I said, day 3 is the viremia in this model we measured. Even up to 48 hours delay, it's substantially reduces the viremia in the treated animals. So this is very good. And then even with a single dose of this compound in the AG129 mouse model, you can see, this is the after infection, immediate treat at this doses, you can see a reasonable dose response, and a single dose can even inhibit the viremia by tenfold. And in the lethal model, it 100% protects the mice from death. So it's all look very good. And then even before, when we did the 1-week preclinical toxicity study in the rats, we received the NOAEL, no observed adverse effect level, continuously 1-week treatment. But in order to move to the clinics, we have to do the 2 weeks of toxicity, both in rats and the dogs. And there, when we found a tremendous toxicities in the 2nd week of dosing. So that was why this compound was terminated. And Novartis made kilograms of those, was really lined up trying to go to the phase 1 trial. But that was just a lesson of for the nucleoside, because this will still be pursued in the field, I'm pretty sure. So now, to summarize nucleoside, the challenge is really vividly exemplified the toxicity in vivo. It comes very late, because if you do a lot of in vitro profiling, they all look good. There is no signals. So that's really the challenge. And also for the nucleoside, it's very hard to do the SAR, because you're really modifying one thing that potentially can affect many functions of the compound, right? Cell penetration, conversion, etc. So, but the opportunity remains open, because compared with previous success of HCV, HBV, and HIV, those are more chronic diseases. You need to treat them much longer time, and then the toxicity is more obvious in those indications. But for dengue, we're talking about a week or no more than 10 days, right? So in that sense, we have advantages of the potential of using the nucleoside as the treatment for the acute. And then the resistant profile, because it's a nuke, and it's really very difficult for the virus to confer resistance. And also because it's a nuke, and then it's really very much like to have a broad spectrum of antiviral, not only limited to all the 4 serotypes, but also can cross over to other, flaviviruses, related viruses. So I think that's the example I just want to discuss as the nucleoside. To tend to the second example, which I went through when I worked for Novartis, which is the rational design for the methyltransferase inhibitor. So as we know, many viruses, including dengue, the flaviviruses, the 5' end of the genome, they all have these cap structure, which really mimics our messenger RNA. The function is really to evade the immune system detection, and it's to enhance the translation of the viral proteins. So those are the 2 main functions of the cap. That's why the virus mimicking them. So now, because flavivirus replication does not go to nucleus, so the replication occurs in our membrane. So they don't get to use the opportunity to use the host, a lot of capping enzyme, which reside in the nucleus, right? So then over the evolution, the virus evolved to encode their own methyltransferase. So the NS5, the N-terminal domain of the non-structural protein 5, that can perform the 2 dual methylation. Here, methylate the N7 of the cap and 2' methylation. And they all use this S-methionine indicated here, this structure, use this to donate this methyl group to the N7 and to the 2'. So we previously showed that this NS5 has this activity biochemically and virologically to demonstrate that. So it will be, if you don't have this method group, the dead virus. We know that is essential for the virus to survive. So that will be a reasonable target. So when we saw the crystal structure of the West Nile and compare with other flavivirus methyltransferases, so you can see this is the ribbon representation, and you can see this is the method donor as adenosine methionine, and the surface representation, you can see this is the donor is over there, the method donor, and then we identify a unique pocket, hydrophobic pocket, highlighted in yellow here, and this pocket is conserved among West Nile, dengue, and the yellow fever. You can see all these, and then so the question is, can we use the byproduct after the methylation? So this methyl group is donated, and then it becomes the compound called S-adenosine homocysteine, right? So can we use this unique pocket to design compound so that we can occupy this pocket and then connect it to the product of this reaction? So that was the idea. So again, the S-adenosine homocysteine, the byproduct after the methyl has been donated. And can we design something like that over here? Right. So working with the chemist, based on the S-adenosine homocysteine, we modify this R group. So the R group, if it's a hydrogen, that's the authentic S-adenosine homocysteine, the byproduct of it. And if you link R group with different aromatic rings, so let's just focus on compound 10 with this aromatic ring attached to this R group. And then if you look at the enzymatic activities, you can see we're testing against the dengue methyltransferase N7 methylation inhibition, the 2'-O methylation inhibition. And then we're also testing the human RNA methyltransferase and human DNA methyltransferase to look for the specificity of this compound. So using the starting point, which is the byproduct of all the methylation, including the human methylase. So you can see it inhibits all the methyltransferases. Doesn't matter it's host or from the virus, right? That makes sense. But now, if you have compound 10, which have this aromatic ring, you see remarkably, not only the potency against the dengue methyltransferase activity has been significantly improved here. And more importantly, this compound lost its activity against the host methyltransferase, demonstrating that this modification is going to confer selectivity to specifically inhibit the viral methyltransferase. So then what we did is we co-crystallized this compound 10 in complex with the dengue methyltransferase. Sure enough, it is really as we anticipated that this aromatic ring snugly fits into this flavivirus methyltransferase conserved pocket. So you can also see there are some slight swinging of the side chains of the amino acid that forming this pocket. So that was one of the examples, but the challenge of this project is really these compounds are not very cell permeable, and then it's very having a hard time, even though you can get into a very potent enzymatic activities, the cellular activities has been very, very poor. So that led to the termination of this effort. So, again, rational design will allow us to select specific inhibitors that selectively target the viral target. Okay, so now, the previous speaker mentioned about the NS4B inhibitor that is currently in phase 1 from 2 companies. The one I work is with Novartis, and then the J&J also have a similar compound, but structurally different. similar mode of action. So at Novartis, we screen using the phenotypic screen, basic replicon or infection assays. We screen the whole Novartis stack, which is about 1 million to 1.5 million at that time. So we identified this compound. This compound, NITD, we named it NITD 618. And this compound inhibits all the 4 serotypes as indicated here at single-digit micromolar. Not great, but it has activity. And it doesn't inhibit West Nile yellow fever, chikungunya, Western equine encephalitis, and VSV. The other viruses, it doesn't inhibit at all. So it's very selective to dengue. So now the mode of action, what we did is we select the resistant virus using dengue replicon. And we identify that 2 mutations in the protein, a membrane protein called NS4B. Contains these 2 mutations, all the selected resistant replicon consistently recover these 2 mutations. So now to validate these mutations, their function in resistance, what we did is we made resistant viruses. So single mutation, because as I say, there are 2 were identified, single mutation and the double mutation. So now let's focus on the lower left. So this is the resistance study. So the black bar is the wild type virus. You can see a very nice dose response inhibition by the compound. And then the single, double, you can see the resistance. So clearly demonstrate that those 2 residues are responsible for the inhibition, for the resistance. So then this is, we published quite a few early version of NS4B inhibitor until we worked on this compound, which is now named NITD 688. The structure is shown at the top. Very similar approach as I mentioned it to you in the previous 3-4 slides. We found after a little bit of MAP-chem, we identified the compound has single-digit to double-digit nanomolar EC50 against all 4 serotypes of dengue. So it was a substantial effort in order to get to cross all the 4 serotypes. Again, using the same approach, we identified that the NS4B contains the resistant mutations. And then we also use NMR, chemical shift perturbation experiments, to clearly demonstrate if it's the Y type. If you Y type 4B protein mixed with the compound, you can see the chemical shift, the red and the red and the black indicated here. If you engineer the resistant mutations into the NS4B protein, there is no longer such shift, really direct indicating there is a direct interaction between the compound and the recombinant targeted protein, which is NS4B, and also the mutations, the resistant mutations identified from the resistant virus that can confer the loss of binding to the compound. So now, in the animal model, we showed that immediate treatment in the AG129 mouse model, you can see a 10 mg per kg BID treatment versus a 30 mg per kg BID treatment. There is a dose response. And if you use the QD dosing, 1 dose per day, you can also show a very good dose response. And if you dose higher, the BID, the therapeutic viremia reduction is even stronger. So we also show you can delay the treatment as indicated on the right. It worked very well in the animal model. So now, if you look at the PK in both rats and the dog on the top, they have a pretty good profile. And based on these data, if you do a 7-day simulation of human dose, 35 mg dosing, and on the lower right side, 150 milligram dosing for 7 days, as indicated here, the trough of the concentration exposure in the human is predicted to be way higher than, in this case, if we do 35 mg, it's way above the 3 times EC50. And then if we dose 150, it's over way above the trough of the exposure of the compound is way above the 10 times of the EC50. So I think this is what we are going, and this compound, it just finished phase 1 and it's looking good, and very exciting program to move this forward. And I think, you know, I will ask the organizer, I have a very interesting want to discuss, maybe 5 minutes. I can wrap up, or you want me to stop here to start the Q&A?
Difficult question. I would actually love to hear on the host targeting, because that's a very different approach from where we in the ASAP are. Yeah.
Okay, so I
if
try
that's
to
okay with everyone, yeah.
Okay, so now I think it's important to talk about this project because we feel the effort has been repeated in many occasions by many groups and many institutions. So I thought it's very, very important to reiterate this type of. So when we did the phenotypic screen, we identified one compound as indicated here on the top left. Very potent, 2.4 nanomolar against dengue. And all the 4 serotypes, they are all in the, you know, single digit nanomolar. And it's not toxic at all. So now, it doesn't want to advance this stuff. Okay, yeah. So you can see here against the other viruses, yellow fever, West Nile, BSV, even, you know, the Western equine, the alpha viruses, it inhibits very well. You know, here, a little bit reducing potency, double-digit nanomolar, but it's still very, very decent. Now, the mode of action, what we identify after a lot of effort by making the biotinylated compound and do pull down, and then the mass spec, and we identify this compound binds very strongly to the DHODH, which is the enzyme involved in the pyrimidine pathway, synthesis pathway, right? So then we expressed this enzyme using the E. coli system as indicated in panel A, and we can direct the binding of the analogs of this series. You can see clearly binds the ITC curve, shows very nice. And then we used the Brequinol, which is known previously, you know, bind to this protein as a control. So it shows very well binding. Now, to further demonstrate the pyrimidine synthesis pathway is involved in this antiviral activity, we did the rescue experiments. So here, what we are doing is we're doing the antiviral assay, but in the medium, side by side, we're adding different concentrations of uridine into the medium. So if the compound is really inhibiting the pyrimidine pathway, by adding these uridine is going to restore or diminish antiviral activity, right? So now, if you look at the black bar here, this is without uridine addition. So it shows, as expected, antiviral activity. But if you have the uridine added, you can see a dose restoration of the viral replication. So again, that really just demonstrates the pyrimidine pathway is involved in this antiviral activity. So now, when we move into the animal model, the AG129 animal model, here, the panel A shows the exposure of this, you know, different routes of the dosing of this compound. And I showed you before, in dengue, this compound is single-digit nanomolar inhibitor. So, you know, it's like 3 nanomolar EC50. So it's way above the exposure, right? The exposure is way above the EC50. So you would expect antiviral activity. But in reality, when you measure the viremia, there is none, even you dose at the very high, 30 mg per kg. So now, it really, at the beginning, we really scratch our heads and say, hey, what's going on? Here, it has this activity, exposure looks good, but in vivo, it doesn't have activity until our PK group started to the uridine concentration in the blood. You can see it really doesn't matter. This is without the treatment, and this is with treatment. You can see a reduction, but the treatment is never able to bring it down to below 6 micromolar, which I showed you in the previous slide. If you add 5 micromolar uridine here in cell culture, it's going to reduce the potency of the antiviral activity. So that really explains, we think that the animals will use the salvage pathway from the food they take, and these will have pyrimidine derivatives, and that's going to diminish the antiviral activities of this kind of. So now to conclude, I think that the few examples I showed is really both viral and the cellular targets can potentially be used for antiviral. And then targeting the host target, it has extra barriers. We just have to be careful, toxicity, and achieve the in vivo activity. And I mentioned that our Novartis compound, NITD 688, and we know that, and also similarly, J&J also is an NS4B inhibitor, and they're also very exciting compound at the phase 1. And then I think for combination therapy, effort is still needed to identify the combo candidate for combination therapy. And lastly, I would just thank the Novartis team when I worked there for almost 8 years, a great team to work with. And through the AViDD, we reconnect again, trying to continue this journey. And I will stop here and thank you for your attention.